Hi everyone, let's learn about different types of selective media. These are the media that will inhibit the growth of unwanted microbes and it will allow the growth of specific microorganism which is specifically required to be isolated. So this medium will contain basal media which contains the carbon, nitrogen, salts or water, and then it will contain the additional elements or nutrients that will specifically inhibit the growth of other microbes and will allow the growth of specific organism which is to be identified or isolated. So these nutrients are selective agents, that is sometimes in some media there will be bile salts, some requires dyes, in some antibiotics or high salts. Distilled water and agar. If the solid medium is required, then agar is required. Let's see the examples of the media that are used as selective media. These are MacConkey Agar, Mannitol salt agar, XLD agar, Thyromartin agar, Hyoctone enteric agar, Cetrimide agar, Scurose medium, Bismuth sulfate agar, Lowenstein-Jensen medium, Tellurite blood agar. MacConkey Agar. This medium is used as both selective and differential medium. It is selective for specifically gram-negative bacteria and inhibits the growth of gram-positive organisms. And it is differential for lactose fermentation, in which the lactose fermenters will appear pink or red colonies, while the non-fermenters will appear colorless. The composition of this medium: peptones, lactose, bile salts, sodium chloride, crystal violet, distilled water, agar, and neutral red. So why these components are added for the preparation of MacConkey Agar? What is the purpose? Peptones are added because they are the source of nitrogen in the form of amino acids and peptides for bacterial growth. Lactose are the sugar for fermentation, as it is also used as a differential medium. So lactose fermentation will decide either these are lactose fermenters bacteria or non-lactose fermenters bacteria. Bile salts are added to inhibit the gram-positive bacteria. As it is selective for only gram-negative, so it will inhibit the growth of gram-positive bacteria by the addition of bile salts. Sodium chloride, it maintains the osmotic balance in the medium. Crystal violet, it inhibits the gram-positive bacteria. Again, crystal violet is also added to inhibit the growth of any remaining gram-positive bacteria. Distilled water is the solvent for medium preparation. Agar is the solidifying agent. And neutral red, which is the pH indicator that turns red in acidic conditions when there is lactose fermentation. So the lactose fermentation will turn the colonies into pink color because of this neutral red. Mannitol salt agar. Mannitol salt agar. It is used as both selective and differential medium. It is selective for halotolerant Staphylococcus species. Halotolerant because Staphylococcus can tolerate the high salt environment. And differential through mannitol and phenol red. When Staphylococcus aureus ferments mannitol, they turn into yellow color, while non-fermenters of mannitol will be in red or pink color. The mannitol salt agar is composed of peptones, beef extract, sodium chloride, mannitol, distilled water, agar, and phenol red. Why these components are added? Peptones in the form of casein and meat provides nitrogen, vitamins, and growth factors. Beef extract provide additional nutrients and growth factors. Sodium chloride is basically the selective agent that provides the high salt environment that inhibits non-halitolerant bacteria. It means that it will allow the growth of Staphylococcus while it will inhibit the growth of other bacteria. Mannitol, fermentable carbohydrate used to differentiate mannitol fermenting staphylococci. So mannitol is basically the carbohydrate which will be fermented by Staphylococcus aureus to produce the colorful colonies. Distilled water, as it is the solvent and used for medium preparation. Agar, which is the solidifying agent, and phenol red, which is the pH indicator. This phenol red, it will turn yellow under acidic conditions from mannitol fermentation. Xylose lysine deoxycholate agar, also known as XLD agar. This medium is used as both selective and differential. It is selective for gram-negative enteric pathogens like Salmonella and Shigella, and their colonies are differentiated by detecting xylose fermentation, lysine decarboxylation, and hydrogen sulfide production. The composition of this medium composed of sodium thiosulfate and ferric ammonium citrate, xylose, yeast extract, sodium chloride, sodium deoxycholate, distilled water, agar, lysine, and phenol red. Why these components are added and which one act as selective and which one act as differential? Sodium thiosulfate and ferric ammonium citrate. It detects the hydrogen sulfide production, creating black centers in Salmonella colonies. Xylose, together with lactose and sucrose, these are the fermentable sugars. Yeast extract, it provides nutrients, nitrogen, and vitamins. Sodium chloride, it maintains the osmotic balance. Sodium deoxycholate, it inhibits the gram-positive bacteria for selectivity, so it will allow the growth of gram-negative enteric pathogens. Distilled water for medium preparation. Agar is the solidifying agent. Lysine is added for decarboxylated by Salmonella after xylose runs out, so it restores the alkaline pH and turns it into the red color. Phenol red is the pH indicator that will turn yellow under acidic conditions and red in neutral or alkaline conditions. Thayer Martin Agar. This medium is selective for isolating Neisseria gonorrhea and Neisseria meningitidis from clinical specimens with mixed flora. It is composed of chocolate agar base, vancomycin, colistin, nystatin, distilled water, agar, and trimethoprim. Chocolate agar base provides essential nutrients including factor 10 and factor 5. Vancomycin, it inhibits gram-positive bacteria. Colistin inhibits most gram-negative bacteria except Neisseria. So it will inhibit the growth of other gram-negative bacteria, but it will allow the growth of Neisseria species. Nystatin or amphotericin B, it inhibits fungi, so the fungal growth is inhibited. Distilled water for the preparation of medium. Agar is a solidifying agent. Trimethoprim, it inhibits the Proteus species and prevents the swarming. Pectoine enteric agar. This medium is used as both selective and differential, which is used for isolating Salmonella and Shigella from clinical and food samples. It is composed of lactose, sucrose, and salicin. bromothymol blue and acetylfuchsin, sodium thiosulfate and ferric ammonium citrate, bile salts, distilled water, agar, and peptone. Lactose, sucrose, and salicin, these are the fermentable sugars to differentiate fermenters from non-fermenter colonies. Bromothymol blue and acetylfuchsin, these are the pH indicators for detecting acid production. Sodium thiosulfate and ferric ammonium citrate, these detect hydrogen sulfide production, which is in the form of black centers in Salmonella colonies. Bile salts inhibits the gram-positive organisms and many other intestinal flora. Distilled water, these are the solvent for medium preparation. Agar is the solidifying agent, and peptone, these provides nitrogen and amino acids for growth. Cetrimide agar. This medium is used for isolation and identification of Pseudomonas aeruginosa from clinical and non-clinical specimens. It is composed of cetrimide, magnesium chloride and potassium sulfate, sodium thiosulfate and ferric ammonium citrate, glycerol, distilled water, agar, and peptone. Cetrimide is the selective agent that will inhibit the growth of other bacteria while it will allow the growth of Pseudomonas aeruginosa. Magnesium chloride and potassium sulfate, it enhances the pigment production like phyocyanin and fluorescein. Sodium thiosulfate and ferric ammonium citrate, it detects the hydrogen sulfide production in the form of black centers in Salmonella colonies. Glycerol is the carbon source that supports pigment production. Distilled water is used for the medium preparation. Agar is the solidifying agent. And peptone, it provides nitrogen, carbon, vitamins, and minerals for growth. Scirose medium. This medium is used to isolate Campylobacter species, particularly Campylobacter jejuni from stool specimens. It is composed of blood agar base, lice horse blood, vancomycin, polymyxin B, distilled water, agar, and trimethoprim. Blood agar base provides essential nutrients and supports bacterial growth. Lice horse blood enrich the medium for fastidious organisms. Vancomycin, it inhibits the gram-positive bacteria, so it will allow the growth of Campylobacter species. Polymyxin B, it inhibits many gram-negative bacteria. Distilled water, solvent for preparing the medium. Agar is the solidifying agent. Trimethoprim, it inhibits the Proteus species and some coliforms. This medium is used as selective for isolating Salmonella species, especially Salmonella typhi from clinical, food, and environmental specimens. It is composed of bismuth sulfite, brilliant green dye, ferrous sulfate, peptone, distilled water, agar, and glucose. Bismuth sulfite, it inhibits gram-positive and many non-Salmonella gram-negative bacteria. So it will inhibit the growth of other bacteria while it will allow the growth of Salmonella species. Brilliant green dye, it further inhibits the gram-positive and coliform organisms. Ferrous sulfate, it detects the hydrogen sulfide production, resulting in black or greenish colonies. Peptone, it provides nutrients for growth to the bacteria. Distilled water, it is used as a solvent for preparation of medium. Agar is a solidifying agent. And glucose is the fermentable sugar source. Tellurite blood agar. This selective medium is used to isolate Corynebacterium diphtheria from respiratory and other clinical specimens. It is composed of defibrinated sheep blood, blood agar base, potassium tellurite, distilled water, and agar. Defibrinated sheep blood, it supports the growth of fastidious organisms and help observes the hemolysis. Blood agar base, it provides essential nutrients for bacterial growth. Potassium tellurite, it is the selective agent that will inhibit most normal flora and it will allow the growth of Corynebacterium diphtheria. Distilled water, it is used as a solvent to prepare the medium, and agar is the solidifying agent. Lowenstein-Jensen medium, also known as LJ medium. This medium is used to isolate and cultivate Mycobacterium species, especially Mycobacterium tuberculosis, from clinical specimens. It is composed of whole eggs, potato flour, glycerol, asparagine, distilled water, malachite green, and mineral salts. Whole eggs provide nutrients and solidify upon heating. Potato flour, it supplies starch and additional nutrients. Glycerol enhances the growth of Mycobacterium tuberculosis. Asparagine acts as a nitrogen source. Distilled water is the solvent for mixing components. Malachite green, this is basically the selective agent that inhibits the most other bacteria except the Mycobacterium species. While the agar is not used because the solidification occurs through the excoagulation. And mineral salts like magnesium sulfate, sodium citrate, potassium phosphate, they maintain the pH and support the growth. This was all about the different types of selective media. Don't forget to watch the series of videos on each medium in which the type of colonies grown on these medium will be explained.